Okay, well, good afternoon, everyone. My name's Ben Hopkins, as Jassim said. I'm a postdoc in Archam Cops Group at UC Davis, and thank you so much for the invitation to speak today, and thank you so much to the organizing committee, and Jassim's been very kind, walking me around and taking me to all of my meetings, so thank you very much personally to you. So clearly one of the central goals of evolutionary biology is to explain the processes that generate this staggering diversity of forms that we see across the natural world. And central to understanding this diversity is understanding the process of evolutionary innovation, the capacity of the evolutionary process to produce traits that are entirely new. So we find evolutionary innovation at basic level of biological organization, from new lineage-specific proteins to new cell types to new organs and morphological structures. And it's really this process that my work tries to understand. What are the genetic, molecular, and cellular pathways through which the evolutionary process innovates? Now, reproductive traits are perhaps the aspects of organismal design that evolve most rapidly. And it's here that we frequently see new structures, new plumages, and other morphological features evolving. But in reproductive traits, we also frequently see the evolution of new molecules that regulate aspects of pre- and post-copulatory reproductive success. So, for example, we see the evolution of new pheromones, new cuticular hydrocarbons in different Drosophila species. And then we also see innovation in the proteins that males transfer to females during mating. So mosquitoes, fruit flies, and moths have evolved these novel, non-homologous proteins that stimulate a variety of post-mating changes in females. So today I'm going to tell you two stories from my recent work, both of which focus on evolutionary innovation in Drosophila reproductive traits. So the first focuses on morphology, and here I'm going to tell you about my work on the developmental genetic mechanisms through which a novel male-specific trait called the sex chrome evolved. And then the second focuses on function, and here I'm going to talk to you about my work on the evolution of a lineage-specific seminal fluid protein. So we'll start with our morphology story. Now, one of the most compelling models that we have for the evolution of novel traits is Gunther Wagner's idea of the individualization of serially repeated homologs. So serial homologs are repeated characters within the same organism, such as duplicated body segments or different limbs or different digits on a hand. And under Wagner's model, the idea is that a subset of those homologs diverge from the others to generate something new. So, for example, the final segment in centipedes is modified to build these larger defensive structures. In apes, the forelimbs and the hind limbs diverge from one another to give rise to arms that end in hands and legs that end in feet. And then on those hands, we have further individualization because we have our thumbs that have diverged from our fingers. And so in Wagner's model, the development of any character is underpinned by a three-tier genetic logic. And in the middle tier, we have a character identity network, or CHIN, that takes in positional information and uses that to activate a downstream network of realizer genes. And it's these realizer genes that actually build the character itself. And the modification of insect wings provides a really neat example of this. So the ancestral state of winged insects is to have two pairs of serially homologous wings. We have the forewings and we have the hindwings. And these wing pairs are essentially identical to one another. So in this case, we can think of the development of each wing pair being regulated by equivalent chins that activate equivalent gene networks that build morphologically equivalent wings. But in Diptera, the hindwings are transformed into these club-like structures called halteres. And the halteres function as gyroscopes that help flies to maintain their stability and balance during flight. And so in this case, the chins that control the hindwing and the forewing have individualized, which is to say that they've undergone some regulatory divergence that makes them distinct from one another. And that decoupling, that regulatory decoupling, allows the downstream gene networks that they control to evolve independently. So in this case, we have a forewing chin that activates a wing building network and a hindwing chin that activates a haltere building network. And this isn't just some kind of weird fly-specific effect. We see something very similar in beetles, except in this case, it's the forewing chin that's individualized and instead of a haltere, we have something called the elytra, which are these hardened protective casings that protect the hind wings. And so I think this is a really neat conceptual model, but we don't really have a very good sense of how this works in practice. And specifically, we don't have a good understanding of the genetic mechanisms through which chins individualize from one another, nor of the mechanisms through which these chins rewire downstream realizer networks that actually build the traits themselves. So these are questions that I've spent the last few years with Artyom Kopp studying, and we've been using the Drosophila sex comb for this. So this is a male-specific organ found on the foreleg, specifically on the tarsus, which is this lower portion of the leg down here. And these sex combs are used in mating. So we can see the male here using his sex combs to grasp the base of the abdomen of the female before mounting and attempting copulation. But when the sex comb is compromised, as we see in this video, the male fails to maintain his grasp, basically just slips straight off the back of the female. And as a result, the probability that the male successfully copulates is massively reduced. Now, given that the sex comb is so crucial in Melanogaster, what's surprising is that it evolved pretty recently. So as a result, the sex comb is only present in a fairly small group of Drosophila species. So if we look at a fairly close relative of Melanogaster, such as Drosophila willistonii, which is shown here, we don't see any sex combs. But then in the lineages that do have them, the sex comb has undergone this explosive diversification in morphology across species. So in Ficus phyla, for example, which is shown here, the sex comb has massively expanded to occupy much of the tarsus. Now, the key feature of the sex comb are these individual bristle-like structures that we call teeth. And these teeth are serially homologous to mechanosensory bristles. The mechanosensory bristles are a very ancient class of sensory organ that's found right across insects. And in the male and female tarsus, these mechanosensory bristles are arranged in transverse bristle rows. So we have a row here at the top, as shown by this blue line, another below it, and so on and so forth. But then in males, the most distal TBR is heavily modified to become the sex comb, and those modifications are threefold. So first, we have this transformation from mechanosensory bristles to sex comb teeth, and these are much longer, they're much thicker, they're more heavily melanized. We also have an expansion in the number of teeth, and then that whole distal TBR is rotated 90 degrees, such that the sex comb sits in a longitudinal orientation. So it goes from this to this. But for the purposes of this idea of individualization of serial homologs, it's really this bristled tooth transformation that's really of interest. So under the Wagner model, we would predict that for the sex comb to evolve, we would need a new chin to evolve in the most recent common ancestor that forces a regulatory separation between the different bristle rows in the leg. And that's exactly what previous work from the Kopp lab has found. So what they've shown is that the gain of the sex comb coincides with the gain of expression of a gene called double sex in the developing sex comb bearing region. And as genes go, double sex is particularly interesting for two reasons here. So first, it's a transcription factor, which means that it regulates the transcription of other genes, often many other genes, determining whether they're turned on or they're turned off. So in that sense, the gain of double sex expression could bring with it the activation of a whole set of new target genes. Then the second reason that double sex is significant here is that it's the primary effector of sexual differentiation in Drosophila. So in this species, sex is determined at the level of the individual cell rather than through circulating sex hormones. And the way this works is that the number of chromosomes determines how the gene double sex is spliced. So we get male-specific isoforms of double sex when few X chromosomes are present, and a female-specific isoform when multiple X chromosomes are present. And it's these sex-specific isoforms of double sex that instruct cells to follow either male-specific or female-specific differentiation trajectories. So it's ultimately the expression of double sex that ensures that the sex comb is a male-specific trait. So if we look in the developing leg of Drosophila species that have sex combs, such as Pseudo-obscura, which is shown here, we find double sex expression in the cells that will eventually give rise to the sex comb, or in this case, combs plural. This is a species that has two sex combs. But then if we look in species without sex combs, such as in Drosophila willistoni, we find no evidence of double sex expression. Now, we also know that the ancestral chin involves a different transcription factor called SCR. So in all of these bristle rows, it's SCR that relays spatial cues into the activation of the bristle building gene network. So that's in all of these rows shown in the blue rectangle. But then in the sex comb, the chin has been modified by the introduction of this gene double sex. And specifically what happens is that double sex establishes an autoregulatory loop with SCR, where each gene elevates the expression of each other. And so this means that in the comb-bearing region, you have kind of two differences from the rest of the leg. You have the novel expression of double sex, and you also get much higher protein abundance of SCR than you do in other parts of the leg. Right, so this is the new individualized chin that gives the most distal bristle row the potential to evolve independently from those other bristles. But the question that I was interested in was, well, now what? You know, how is it that this new chin is rewiring downstream gene expression to turn what was a mechanosensory bristle into a sex comb tooth? And to understand that, we need to think about the development of these individual bristles. So any one of these mechanosensory bristles or sex comb teeth is a multicellular organ composed of 4 distinct cell types. So we have a shaft cell, and it's this cell that builds that projection. We have a socket cell that anchors it, a neuron, and a sheath cell, which connects the neuron to the shaft. And these 4 cell types are generated from a shared progenitor cell, called a sensory organ precursor, via successive rounds of cell division. And this then seems to be a pretty general blueprint for building different classes of sensory organ. So variants of these cell types are generated through very similar patterns of division in the sex comb, in the mechanosensory bristles, in the chemosensory bristles, and several other different sensory organs. And so given the structure of this cell lineage, it follows that to turn a mechanosensory bristle into a sex comb tooth, there must be some genetic rewiring going on within this shaft cell lineage. And broadly speaking, we could imagine 2 quite general mechanisms that might drive one of the key features of the sex comb, which is this massive increase in tooth size. So the first is earlier specification of sex comb cells. So basically, if you can just establish the identity of the shaft cell earlier in development, maybe you give the shaft longer to grow and it can therefore reach a greater size. Alternatively, perhaps there are differences in the growth rate between the comb and the mechanosensory shafts. So maybe the shafts of the comb grow much faster, which allows them to ultimately reach this greater size. So we began by testing this specification hypothesis. And our idea here was to simply ask, when do we first detect the transcription factors that ultimately establish the identities of the shaft cell? And what we found is that these transcription factors are expressed in the sex comb cells much earlier than they are in the mechanosensory bristles. So in this zoomed in image here, you can see that we have purple suppressor of hairless staining present only in the sex comb at this early developmental time point. So expression of suppressor of hairless in the comb, but not in the adjacent transverse bristle rows. So the cells of the sex comb appear to be specified earlier than the cells of the mechanosensory bristles, but can this then account for the morphological differences between them? Well, in females, the homologous bristle row to the male sex comb looks completely identical to all of the other bristle rows. So I'm highlighting the position of that here with this arrow. So if earlier specification gives us these larger, thicker bristles in males, then we shouldn't see earlier specification here in females. Except that we do. So in the females, we see earlier specification of the bristle cells that are homologous to those of the male sex comb. And yet in this case, there appears to be no associated morphological consequences of this. So earlier specification doesn't itself seem to be sufficient for this bristle to tooth transformation. So what then about differences in the rate of shaft growth? Well, to assess this, we expressed an RFP tagged form of actin in the developing bristles and measured the lengths of the shafts at different time points. And what we found is that there's no shaft growth in either the comb or the mechanosensory bristles prior to 24 hours after puparium formation. But then from 26 hours, we start to see growth in both the comb and the mechanosensory bristles simultaneously. And this growth then quickly ramps up such that by 38 hours, the shafts are almost fully grown in both the sex comb and the mechanosensory bristles. And when we look at the difference in length between the mechanosensory and comb shafts, we find that that difference increases between 26 hours and 30 hours, and that difference increases again through until 34 hours. So what this means is that we have an acceleration in growth rates in the sex comb teeth, such that with each hour, they're getting progressively and relatively longer than the mechanosensory bristle shafts. And that size difference then drops at 38 hours as sex comb growth starts to slow. So if we go back to this original model, we can now add in that this newly evolved sex comb chin regulates the bristle to tooth transformation, in part by accelerating the rate of shaft growth. But that's just the developmental mechanism. The next question we are interested in is, what are the genetic changes that drive this acceleration in shaft growth? And to understand this, we needed to find a way of comparing gene expression between differentiating shaft cells in the sex comb and in the mechanosensory bristles. And for this, we turned to single-cell RNA sequencing. Now, in order for us to use this technique, we had to first find a way of dissecting out the individual tarsal segments that actually carry the sex comb, and to do this in developing pupae. And just to give you a sense of how challenging this is, we've got a pupa shown here on the screen, and we've carefully peeled away its outer casing. And we have the foreleg tibia shown here in purple, and then each of the 5 tarsal segments. And the position of the sex comb is shown with that blue circle. So we were using really super fine scale dissections to cut just between these 2 dashed red lines. So this is a very small piece of tissue that we're isolating here. Basically took us about a year to come up with this dissection protocol. Now, once we had about 70 of these segments dissected out, we would dissociate the tissue into individual cells using a kind of cocktail of enzymes, and then use these cells as the input into a microfluidics-based single-cell RNA sequencing platform. And the idea here is that individual cells are encapsulated within unique reaction vesicles. And these vesicles contain all of the reagents required to turn mRNA into cDNA and to tag those transcripts with a unique barcode for that particular cell. And it's that unique barcode that then allows us to assign in our sequencing data individual transcripts to individual cells. And we repeated this process for 6 different developmental time points, ranging from 12 hours to 36 hours. And as we've already discussed, this is covering many of the key stages of sex comb development. So from the final cell divisions through the rotation of the comb, this going from a transverse to longitudinal orientation, to then covering much of the growth of the shafts themselves. Now, for those who aren't familiar with single-cell sequencing work, the outcome of one of these experiments is a count matrix like this. So this gives us a readout of the number of transcripts detected for each gene in each of some 10,000 cells that we sequence in any one sample. And one of the challenges of these data is to then identify the cell types that each sequence cell corresponds to. So to be able to say, for example, in this case, that cell 1 is a hemocyte and cell 2 is a neuron. And one of the ways we can do this is to cluster cells based on their similarity to one another, with the ideas that cells that form a kind of cohesive cluster are likely to belong to the same cell type. And we can then look at the sorts of genes that are enriched in these clusters and use that as a way of identifying the cell types that those cells correspond to. But really, we were going into this work pretty blind. So when we get our first data sets back, we were met with all of these different clusters, most of which we couldn't identify. So the first part of this project was to assemble a developmental cell atlas of this tissue, which we published last year. And in the course of this work, because we were sequencing not just the sex comb, we were getting the broader first tarsal segment, we got these other cells popping out, and a number of interesting side stories emerged from this. So for example, we discovered a new cell population that's labeled here in green, and this wraps itself around the bundles of nerve fibers, which are stained here in purple, that run through the length of the leg. And this cell type seems to be involved in building the outermost layer of the nervous system, the neural lamella. And then we were also able to detect different subclasses of chemosensory neurons. So whereas the sex comb and mechanosensory bristles are each innervated by just a single neuron, every chemosensory bristle is innervated by 5 different neurons. We have a mechanosensory neuron and then 4 different sensory neurons. And it's these chemosensory neurons that are involved in mate detection, for example. So these are actually tasting neurons. They respond to different pheromones. And one of the things that we were able to show is that each of these different neuron classes expresses its own unique combination of transcription factors. So in these confocal images, we're looking at each of the neurons in two individual chemosensory bristles. And here we can see that in both cases, all four of the chemosensory neurons express a transcription factor called prospero. But then one neuron type expresses nervy and another neuron type expresses forkhead. And so by piecing these different expression patterns together, we're able to assemble a hierarchical combinatorial transcription factor code that seems to uniquely specify each of the different sensory neurons within one of these chemosensory bristles. That's a little bit of a tangent because for the purposes of this talk, really the key output of the study was our identification of marker genes that allow us to identify each of the different cell populations within sexcomb teeth, mechanosensory bristles, and chemosensory bristles. So having identified shaft cells from these different organ types, the first analysis we ran was very simple, just to compare their transcriptomes and ask how many differentially expressed genes do we see between them, and at what time points do we see the greatest difference. And we found several hundred differentially expressed genes, with the number increasing as we proceed further through cell differentiation. And as a reference, we also ran the same analysis comparing the mechanosensory to chemosensory shafts. And in this case, the transcriptomic differences appear to be a little more modest. But of course, the key question is, well, what are these gene expression differences? And at this point, I think it's worth thinking about what forms of gene expression change we might be seeing. And there are three broad modes of change that could be possible. So the change might be qualitative, where genes that are absent from mechanosensory bristle shafts are co-opted to be expressed in the sexcomb. So genes that might have ancestrally be expressed in non-sensory organ cell types. And this is the sort of thing we could imagine happening when we introduce a new transcription factor into the tissue. So as I said earlier, you could imagine double sex bringing with it a whole host of new target genes. But alternatively, the changes in expression could be quantitative, where the level of expression of some gene that's expressed in both the sexcomb and the mechanosensory bristles is changed. The level of expression is changed between them. Or it could be heterochronic, where the time frame over which a gene is expressed is changed between the cell types. So you could imagine, for example, some genes being turned on earlier in the sexcomb cells than they are in mechanosensory bristles. Now, in terms of the qualitative differences, we see essentially no evidence of this. So across our 800 or so differentially expressed genes, essentially every one shows some expression in the mechanosensory bristles as well. The one exception to this is a gene called BLAC, which we see at pretty high levels in sexcomb shafts from about 20 hours onwards. And then it's entirely absent from the mechanosensory and chemosensory bristles. And this isn't something we just see in our single cell data. So we see the same thing in our HCR in situ. So BLAC is restricted to the sexcomb at 30 hours. And then if we go back at a later time point, if we look at 48 hours, we see that it's still restricted to the sexcomb. And in contrast, BLAC is absent from the developing female tarsus. So this is a gene that seems to have specifically evolved expression in the male foreleg. But our experimental data suggests that BLAC is actually completely dispensable for sexcomb development. So we've screened lots of different trans heterozygotes now that carry various combinations of BLAC mutant alleles. And in every case, sexcomb development looks entirely normal. So if qualitative changes aren't important, then how about these other modes of expression evolution, quantitative and heterochronic? Well, here we find much more widespread effects. So here's one example of this, a gene called peroxidase. Now, through until 30 hours, peroxidase looks like a qualitative gain. It's only expressed in the sexcomb. And if we look at the in situs, we see the same thing. So it looks just like that gene I was talking about, BLAC. So both peroxidase in green, BLAC in purple are fully restricted to the sexcomb at this time point. Then after 30 hours, we can see in our single cell data that peroxidase starts to be expressed in the chemosensory bristles. And by 48 hours, our stainings show that it's expressed in every bristle cell in the leg. So this is an example of a heterochronic shift with expression activated earlier in shaft development in the sexcomb compared to in the mechanosensory bristles. And we see this in several other genes too. So CG4374, for example, is turned on earlier in the sexcombs. And then we see many other genes, including these ATP synthase subunits, which are expressed at consistently higher levels in the sex chromosome. So these cases reflect quantitative shifts in expression. So how does this affect our model? Well, we know that the sex chromosome has evolved by establishing this new chin, and this chin drives accelerated growth in the shafts. And we can now fill in this gap to say that the chin does this by modifying both the level and timing of expression of genes that are also present in the mechanosensory bristles. So this is to say that the chin orchestrates a quantitative and a heterochronic rewiring of a gene program that builds bristles that's otherwise pretty conserved. But the question then, of course, is, well, what are the genes still, and how does this drive the accelerated growth? Well, this is where things became a little more computationally challenging, because we have these 800 genes that show quantitative and heterochronic differences in expression, but how can we then make meaningful mechanistic predictions from that many genes? Well, this is where we turn to a recent method called DPGP. And what DPGP allows us to do is to cluster differentially expressed genes based on their expression trajectories through time. So here we see one of the clusters identified through this analysis. We have individual gene trajectories shown with light gray lines, the cluster mean given as a dark blue line, and then we have these different time points that we sampled on the x-axis. And the way to read this is that larger values reflect greater differences between sex chromosome and mechanosensory shafts, and then smaller values reflect smaller differences. Now, when we take these clusters together, what we can start to see are these waves of gene expression changes through time. So in cluster 1 here, we have genes that peak in their expression differences at 16 hours. And then in clusters 9 and 7, we have genes that show this gradually increasing expression difference as we proceed further through cell differentiation. And we can then use gene ontology analyses to search for particular modules of function that are associated with these individual temporal trajectories. So in cluster 1, for example, we're seeing significant enrichment of terms relating to ribosome production. And from 24 hours, we're seeing this gradual uptick in genes associated with energy production. So ATP metabolism, oxidative phosphorylation, electron transport chain. And then in cluster 7, we see this continuing increase in genes associated with protein synthesis. So if we return back to this model, we can now link this quite general idea of quantitative and heterochronic changes in existing gene programs to the accelerated shaft growth. And that link is that these expression changes act through a range of gene programs that regulate pretty general cellular processes: ribosome production, energy production, protein synthesis. And these changes open up a specific testable prediction. And that's, although energy availability is obviously going to be important if you're building any sort of shaft, we can imagine that sex chromosome development shows this heightened sensitivity to the availability of ATP. And so this is something we're starting to look into at the moment. And the way we're doing this is to use cell type specific RNAi knockdowns to compromise the efficiency with which ATP is produced. So here you can see the result of that when we activate this exclusively in the sex chromosome. We're compromising ATP production in the sex chromosome, and this leads to much shorter, thinner teeth. But the important thing here is going to be that comparison with the mechanosensory bristles around it, and I'm running the crosses back in Davis as we speak, so I don't have the data for that just yet. But there's still a missing part of the puzzle here that I want to talk about, and that's how does the new chin drive these quantitative and heterochronic changes? Well, we have some clues about this from our clustering analysis. So here we've got another one of the clusters that comes out of this, this analysis that shows a peak at 16 hours. And this cluster is significantly enriched for terms relating to the cell cycle and to DNA replication. Now, this is pretty surprising. So we know that by 16 hours, these cells have completed their divisions. So why is it that they're replicating their DNA? Well, to understand this, we need to think about the cell cycle. So in the cell cycle, we usually think of cells replicating their DNA before dividing it between daughter cells during mitosis. But that DNA doesn't have to be divided. We can also have endoreplication, where the cell continues to replicate the genome without dividing. And cells can undergo many rounds of endoreplication, leading to substantial increases in ploidy level. And by repeatedly duplicating the genome, the copy number of individual genes is amplified. This provides a fairly simple route through which you could boost gene expression. And this is exactly what seems to be happening in the sexcomb. So here you're looking at measurements of nuclear area, which is a well-established proxy for ploidy level. And we're looking at shaft cells in the sexcomb in purple in the plots, and the adjacent mechanosensory bristles shown in orange and green. And across all of the time points that we looked at, the sexcomb nuclei were substantially and consistently larger than those in the mechanosensory bristles. And crucially, when we look in the females, we find no difference between the most distal bristle row, the one that's homologous to the sexcomb, and the other bristles. So this increase in ploidy level seems to be a male-specific, sexcomb-specific effect. So we can now fill in this last gap in our model and say that the new chin drives accelerated growth, at least in part by inducing additional genome duplications, and these boost the expression of gene modules involved in energy and protein production. So that's where we're currently up to with this work. And the questions I'm now thinking about are, how does this chin stimulate endoreplication? What's the genetic trigger of this? And then also thinking about the melanization of the sexcomb teeth. So this is perhaps the most conspicuous change in the sexcomb, but is that genetically coupled to changes in bristle growth dynamics? You know, if you just accelerate shaft growth, do you inevitably get this cuticular melanization change? Or is this something that's controlled by a distinct later acting genetic module? And I'll just end this section with two thoughts about the significance of this work. So I think that we're often used to thinking about gene co-option as the driver of novel trait evolution. So to evolve a new trait, you need to activate the expression of a whole host of new genes in ancestrally naive cells. But that's not what we're finding here. Instead, our work is suggesting that you can get these pretty large-scale phenotypic shifts simply through changes in the timing and level of expression of components of pre-existing gene modules. And so I think that this is potentially a fairly simple genetic route through which the evolutionary process can innovate. So that was a story of morphological innovation. I'm going to change gears a little bit now and tell you a story about functional innovation. So mating stimulates behavioral and physiological changes in the females of many different species. And we see this pretty much everywhere we look in insects, including in flies, in thrips, in beetles. And the two most common changes that we see, probably because they're the easiest to measure, are a reduction in sexual receptivity and an increase in egg production. And we know that in many species, these changes are induced by seminal fluid products that males transfer during mating. So it's not just sperm that males transfer to females, it's also proteins and vesicles, lipids, and much else besides. And I think one of the big surprises that's emerged from this field over the last couple of decades is that different species seem to use different seminal proteins to achieve these same phenotypic effects. So the mosquito, Aedes aegypti, uses a protein called HP1, Drosophila melanogaster uses sex peptide, and the moth Helicoverpa uses PSP. And crucially, these proteins are all apparently non-homologous. They are lineage-specific proteins that have evolved entirely independently of one another. And so these phylogenetic patterns raise the question of how and why do seminal regulators of female reproduction evolve and diversify through time. Now, sex peptide is by far the best functionally characterized insect seminal protein. So it's a short peptide, just 36 amino acids long in its mature form. And it's produced in the male accessory glands, which are a pair of glands that branch off from the base of the testes. Sex peptides then stored and trafficked on these lipid-based structures called microcarriers, and these microcarriers are transferred to females along with sperm during mating. And then once inside the female, sex peptide binds to the surface of sperm and enters into the sperm storage organs. And this binding is controlled by a network of other proteins that males transfer in their seminal fluid. Now, once inside the female storage organs, sex peptide is gradually cleaved off the surface of the sperm over the following days, and that then frees sex peptide to bind to a receptor that's expressed in a small population of neurons that innervate the female reproductive tract. And this receptor is called sex peptide receptor, or SPR. And it's through SPR that sex peptide carries out many of its behavioral and physiological effects. So females become more aggressive after receiving sex peptide. They become less receptive to remating. They show changes in their dietary preferences, in their metabolism, in their digestion. And the way we often talk about this is it's as though sex peptide is flipping a switch in females that transitions them from a pre-reproductive to a reproductively active state. And so when we think about the evolution of this system, we have to reconcile these two different things. So on the one hand, we have this apparently very complex machinery that sex peptide works with, the sperm binding protein network, the sex peptide sensing neurons. There's presumably an enzyme that's involved in cleaving sex peptide from the surface of sperm. Then on the other hand, we have this very rapid turnover in the identities of the proteins that regulate female post-mating behavior and physiology. So with that in mind, I wanted to drill down into the evolutionary assembly of this system to explore how sex peptide and its receptor are evolving. So when did sex peptide first evolve? How has it evolved over time? And how does it co-evolve with its receptor? And these are the sorts of questions that we can now start to answer in Drosophila thanks to a recent explosion of genomic data. So thanks to a collaborator of mine, Bernard Kim, and his colleagues, we now have highly contiguous genome assemblies available for several hundred different species of Drosophila, as well as some early branching relatives. So to identify sex peptide genes across the phylogeny, I used a reciprocal best blast approach. And the basic idea here is that we blast the protein sequence of sex peptide from melanogaster against another species. From that, we get some number of hits returned. We can then take those hits and blast them back against the melanogaster genome. And if the top returned hit is sex peptide, then we can classify it pretty comfortably as a sex peptide gene. And if it isn't, then we can probably discard it. And we can then do the same thing for the genes that flank sex peptide to build up these kind of wider syntenic gene neighborhoods. And we can use those neighborhoods to check that our sex peptide genes fall in conserved genomic locations. So the first thing that emerged from these analyses is that sex peptide is restricted to the Drosophilidae subfamily. So that's this portion of the phylogeny shown by the red line up here. So we didn't find sex peptide in mosquitoes or in houseflies or in other acalyptrate genera like the Liriomyza shown here, nor do we find it in the Steganinae, which is one of the two subfamilies of the Drosophilidae. But we do start to detect copies of sex peptide in the other subfamily, the Drosophilinae, and not just in the genus Drosophila, but also in some of these other genera like the Chimamyza, the Lysocephala, the Scaptodrosophila. Now, this very recent origin of sex peptide contrasts with its receptor, which is very widely detected across invertebrates. So we don't just see SPR in Drosophila, we don't even just see it in insects, we see it right the way out to gastropods, such as sea slugs. Now, for the next part of the talk, I need to give you a kind of brief introduction to the Drosophilidae tribe, which is highlighted with this kind of orange box. So the vast majority of the species that we were sampling in this project came from this clade. And the Drosophilidae contains a huge amount of species diversity. So we have the melanogaster and the montium groups, the obscura, zaprionus, immigrans, quinaria, virilis, repleta, and the Hawaiian and Scaptomyza groups. And then in this highlighted region, which contains the melanogaster, montium, ananassi, the obscura groups, this region is known as the Sophophora subgenus. And our data suggests that sex peptide has followed a very distinct evolutionary path within this specific lineage. So remember, this is the lineage that includes Drosophila melanogaster. And that path is distinct in 4 different ways. So first, we see these repeated independent losses of sex peptide outside of the Sophophora. So we have a single loss shown here by this pink line, which covers the Repleta, the Nanoptera, and the Bromelii groups. We then have other losses at the base of the Hawaiian group and in sublineages within the Scaptomyza and the Hirta Drosophila. In contrast, we didn't find a single loss in any of the 100 or so species that we sampled from the Sophophora. Second, we find that sex peptide has undergone these repeated independent rounds of copy number amplification in the Sophophora. So in the Ananassi subgroup alone, we find species with 2, 4, 5, 6, or even 7 copies of sex peptide. So this plot shows the position of those copies, which you can see as kind of clusters of these black lines here within this wider syntenic gene neighborhood. And then we also find independent lineage-specific gene duplications in a whole host of other species within the Sophophora. So in Gununcola, in Takahashii, Ficus phyla, Subobscura, many other species. In contrast, we found only 1 duplication out of 137 non-Sophophora species. And third, we find that there's considerably more variation in sex peptide protein sequence within the Pseudophora than outside of it. So if we look at the distribution of predicted protein lengths, we find much greater spread among Pseudophora sequences, which are shown here in gold, than outside of it. And that variation carries through to different paralogs within the same species. So Bipectinata, for example, encodes a 46, a 49, a 54, 62, 68, and 72 amino acid long copy of sex peptide. And then the final major difference we see is this sharp uptick in the rate of translocation of sex peptide genes. So aside from the cases where it's been lost, outside of the Pseudophora, sex peptide is essentially always in the same genomic location. So I've depicted that here with these black horizontal lines on Muller element D. But then at the base of the Pseudophora, sex peptide is translocated to a new genomic location on the same Muller element. And since then, it's really jumped all over the place. So it's retained in that new position in Obscura, but then it's jumped to a new element in Persimilis. It's translocated to a different position in Ficus phyla and in Suzuki'i and in Takahashi'i and in Eugracilis and in Melanogaster and in several other species that aren't shown here. So sex peptide seems to have this especially dynamic history in the Pseudophora, frequently hopping to new genomic locations. And this again contrasts with what's going on outside of the Pseudophora, where we have just 2 translocations in the 137 species that we analyzed. So drawing together these different axes of variation, copy number, sequence, genomic location, we see this clear burst of evolutionary activity in sex peptide in the Pseudophora, this apparent lineage-accelerated evolution. So what's driving this? Well, at this point, let me introduce you to the 2 major hypotheses that people have historically used for thinking about the adaptive value of sex peptide. So the first we could broadly call the mating signal hypothesis. And the idea here is that females use sex peptide as a signal that sperm are in storage. And this then allows them to tailor their reproductive behavior to the continuing presence of sperm. The second we could broadly call the manipulation hypothesis. And the idea here is that males evolved sex peptide as a way of manipulating female physiology to the male's advantage. So by reducing his partner's sexual receptivity, for example, a male reduces the probability that his partner remates, and as a result, reduces the probability that he'll encounter competition from rival males. And there are some data suggesting that this receipt of sex peptide reduces female lifetime reproductive output. So in pursuit of their own reproductive success, a male potentially is causing collateral damage that reduces female reproductive success. And it's this data that have led to sex peptide being held up as a sort of poster child example of a trait evolving under sexual conflict. And evolution under sexual conflict opens the door to a sexually antagonistic form of coevolution. And the logic here is that a trait evolves that improves a male's performance in reproductive competition. This trait comes at a cost to the fitness of his female partner. This in turn selects for counter adaptations that reduce those costs to females. But in countering the male trait, the female response depresses the fitness of the male, which then feeds back to renew selection on male traits. So in our case, we could imagine that sex peptide evolves to promote male sperm competition performance, perhaps by suppressing female receptivity to remating. Sex peptide suppresses female fitness. This then selects for female responses that reduce the cost of sex peptide, which depresses male fitness, in turn selecting for evolutionary change in sex peptide. And so perhaps we're seeing some of these dynamics play out in the Pseudophora. Perhaps females evolved resistance to sex peptide in this lineage, which then in turn triggered a burst of evolutionary change in sex peptide. Well, if this is occurring, then we might expect the sex peptide receptor to be one of the primary loci at which female resistance evolves, because it's through this receptor that sex peptide induces many of its effects. The sex peptide receptor is kind of the front line. So to test this, we extracted the sequence of SPR from 180 different Drosophila species. And the basic idea of this was to ask, do we see some shift in the evolutionary dynamics of SPR in the Pseudophora? And to cut a long story short, the answer here appears to be no. So we find no duplication of SPR. There's no parallel pattern of copy number amplification like we see for sex peptide. Then we sort of used a whole barrage of different molecular evolution analyses. So we used busted and found no evidence of episodic positive selection acting on SPR, so suggesting that SPR isn't undergoing these kind of bursts of adaptive change. Using ABS-REL, we find no evidence of any individual branches in the phylogeny experiencing positive selection, suggesting, again, against adaptive divergence of SPR. Using FUBAR, we find no evidence of pervasive positive selection across the phylogeny at individual sites in SPR. However, when we use mean, we did find evidence of episodic positive selection at 9 individual sites in SPR, suggesting that maybe adaptive changes have occurred at a very small number of sites in a subset of lineages. But crucially here, the lineages in which those sites we found evidence for positive selection were evenly split between the Psophophora and the non-Psophophora. And there was no individual change that correlated with a burst in the rate of sex peptide evolution. And so bringing all of this together, we don't really find any clear signal of correlated diversification between sex peptide and its receptor. We have this elevated rate of change in sex peptide sequence diversification, translocation rate, copy number amplification, but without any clear corresponding changes in SPR. So the evolution of these two genes seems to be decoupled at these kind of broad macroevolutionary scales. So what does that mean for this hypothesis that sex peptide is evolving through a sexually antagonistic arms race? Well, if this race is playing out, then it doesn't seem to be doing so, from our data at least, through SPR. But that isn't evidence in itself against the manipulation hypothesis. So there could be other loci that are co-evolving with sex peptide. For example, female resistance could be evolving in the neural circuitries that relay the detection of sex peptide to the brain, and the neural circuitries then that coordinate a behavioral and physiological response to sex peptide. Or perhaps sexual conflict just isn't an especially important force in shaping the evolution of the system. So while there is some data suggesting that sex peptide is sexually antagonistic to female fitness, in the past 10 years or so, a series of papers have come out presenting data that suggests sex peptide may actually be neutral or even beneficial to female fitness in a kind of context-dependent manner. And if that's the case, then if there's no conflict, it's perhaps unsurprising that we don't see this signal of co-evolution between the two genes. But there are other non-sexually antagonistic forms of co-evolution that don't involve SPR that may be at play. So ultimately, the activities of sex peptide are driven by its interactions with the female reproductive tract, and we know that the morphology of the reproductive tract evolves extremely rapidly in Drosophila. So, for example, the sperm storage organs of Drosophila bifurca are massively expanded relative to species like Pseudoobscura and Melanogaster. We also see significant evolutionary changes in sperm morphology. So some species have these extremely elongated sperm, as shown in this image on the bottom right here. So that's a single sperm that's been wrapped around the individual that made it. And so we could imagine that these changes in sperm morphology and in female reproductive tract anatomy could select for corresponding changes in sex peptide. So these changes in sex peptide may, for example, influence the kinetics of its binding with sperm, or perhaps longer sperm need higher copy numbers of sex peptide to ensure that there's enough protein available to fully coat them. But the diversification of sex peptide may also not be a product of coevolution at all. And so at the morphological level, we routinely see cases where male structures show much higher diversity than do the female structures that they interact with. So in springtails, for example, males use these modified antennal segments to grasp the antennae of the female. And if we look across species, we find that the morphology of the male antennae evolves very rapidly. So we see all of these different secondary grasping structures appearing and diversifying. But then if we look in the females, we see essentially no corresponding change. We could make a very similar argument here for the sex chrome, where we see this extraordinary diversification of sex chrome morphology, but without corresponding change in the parts of the female the sex chrome contacts. And yet this does seem to be quite a widespread phenomenon. So male diversification in the absence of corresponding female diversification. So there's a paper here by Bill Eberhard where he identifies tens of different cases across different arthropod groups. So maybe in sex peptide, we're seeing at the molecular level something that's been pretty well documented at the morphological level, rapid diversification of a male trait with limited corresponding change in the female trait that it interacts with. And if you're interested, you can check out the paper here, which came out earlier in the year. And with that, I'll say thank you to collaborators and funders and to everyone here for listening. So thank you.